And now we will drastically change the scale of the analysis because we go from farm to genes. So I would like to introduce John McKay, who will talk about gene model redundancy in the context of nitrogen use. So the floor is yours.
All right, thank you. Thanks to the organizers for having me here. And yeah, we'll go a little conceptual and a little reductionist now. But hopefully you see how it connects to everything. So this is one of our research fields in Colorado at 1,800 meters, where we're screening maize for response to drought and nitrogen. This picture was taken when the vapor pressure deficit was 6.4 this summer. So this is also a target environment that the companies don't breed for. This is Iowa in 50 years, according to the NASA climate model. So, and I'll start with the acknowledgements. So I'll talk about mostly collaboration with Corteva and with folks at University of Würzburg, and work that was done by Kirsten Hein, a PhD student in the lab, as well as work from some of these other people. And then funding is provided by these agencies here. So I'm talking more about the inputs in terms of management and the nitrogen problem in crop production, is particularly in U.S. maize production. As you know, U.S. farmers do have a pretty similar management for their maize. They all put down very large amounts of nitrogen to maximize yield, and the government pays a lot of money for that. So it's considered a public good, and has brought us to large food surpluses. But there's the cost to that in terms of runoff, as well as nitrous oxide production from this fertilization approach. So thinking of how do we move from this current U.S. System of corn and soy to a more sustainable system, there's a lot of economic and socioeconomic challenges that I'm not qualified to address, but there's also some genetics we can think about. So this is in collaboration with Corteva, and we're looking at different genotypes as, so basically management by genotype here. This cartoon just shows 3 different modern hybrids, commercial hybrids, and this 55% reduction in nitrogen rate is basically what's needed to create a corn crop that doesn't have a huge carbon footprint. So that's the calculation there. And what you can see is that they're diligently breeding for a very high yield under high N. And I say, the percent of selection that is spent on yield at low N is extremely low in these industrial breeding programs. And so basically, we've shown here now this curve is on the y-axis, and on the x-axis is basically how big the root systems are of crops. And so we see some mechanistic relationship here that bigger root system, genotypes with bigger root systems are able to get that limited nitrogen. And so we'll revisit trying to understand the genetics of these root traits and their uptake. But let's think about this curve a little in terms of evolution. The breeders are working very hard, as I said, applying directional selection to maximize yield at this 100% nitrogen rate. And we think of this function of this curve of G by M as basically evolving mostly by drift, or this trait being neutral in most of those breeding programs. So the percent of the selection that's acting at this level of nitrogen is extremely low. And so just by chance, these perfectly great award-winning hybrids that win the Iowa State Fair differ. They both are great on high end, but one of them, just by chance, is pretty good at making yield at low end. And so that's the concept that we're going with here to look at that. And I just put in a large proposal to try to address this. But now I'm going to tell you the reductionist story. So we looked at this trait of root mass using root pulling force, and we found it's highly polygenic. We found a number of large effect alleles. One is shown here, where we have the B73 in black, and then the alternative allele that gives you much larger root systems under both full and limited irrigation. This gene model, if we then try to figure out what this gene does, we look at the annotation, and it's related to these Arabidopsis genes that are anion channel genes. And there's a family in Arabidopsis of anion channel genes, including the SLAC gene that controls guard cells, but there's also a number throughout the plant that are involved in nitrogen transport and especially nitrogen signaling. And so they kind of play this dual role of transporting the nitrate as well as sending a signal in the plant of the nitrogen status. The challenge, and we created mutants of this and showed that it is this gene model that's affecting the phenotype under both high and low nitrogen. We get differences in the root mass as well as how it responds in terms of the morphology and root tips. Now we want to try to figure out how this gene is working. The challenge is the lack of one-to-one orthology between our model system Arabidopsis and maize. So we can eliminate that it's some of the possible orthologs that it's related to based on sequence similarity. But there are two genes in Arabidopsis that are equally evolutionarily related to, in this case, 7 orthologs in B73. If you look across maize, there's actually each inbred line has a different number of these. So there's a lot of variation in the presence and absence of these gene models. So now to figure out what our gene actually does, we're using the data from Arabidopsis and trying to recapitulate the phenotype and basically use functional similarity as a definition of orthology here, since evolutionary orthology doesn't work across these distances with this much whole genome duplication. I'll just show all of these. We're not going to go deep into membrane physiology here, but it's known what the cofactors are and the behavior, basically, of these genes in Arabidopsis. And the idea is if we were testing the hypothesis that our gene is functionally orthologous to SLA3, that gene in Arabidopsis that's very well studied and a nitrate-specific transporter. So we're going to test whether this allele behaves like this gene model versus others. And to get to the punchline, we were able to assign this to this SLA3 gene model in Arabidopsis. So the compared to SLA2 in Arabidopsis and the other orthologs, these gene models in maize respond to these cofactors and have the same membrane physiology. We do also see some evidence of, and that this is more data basically showing that all of the cofactors give you the same general pattern between Arabidopsis and the maize gene model. And when we compare the two alleles that we originally identified, we also see some difference in their functional effect in terms of their membrane transport. So this is all in xenopus oocytes that were expressing these maize alleles and Arabidopsis alleles and doing this comparative work. So now we think we know what this gene does, but it was, doing this one gene at a time and trying to re-understand all of the data from Arabidopsis, all of the beautiful functional data, and move it into our cereal crops is challenging. And the reason for this, and I think we all know this, but the scope of it is kind of staggering. So there's a whole genome duplication along the maize lineage, and 2 of them along the Arabidopsis lineage, and so that just blows apart how many minutes? 2 to 3, okay. So it's complicated, and the tools, unfortunately, the people that are well-funded to do evolutionary orthology tools, the human genetics community, don't have this problem to the same degree as us, so their tools don't work very well. So we need to make new ones. Just to show you, this is these 3 maize inbred lines. If we go back to the vertebrates, are as different as chicken and human, right? So that's a huge difference. And what we're looking at now is testing this hypothesis or this theory that it is actually these duplicate or redundant gene models that are evolutionarily free to neo-functionalize and underlie both G by E and genotype by management interaction. So I will end there, but these are 3 different stories that are still moving, but we're working under this idea that again, that if we're looking at these functions, part of these functions is evolving under very strong selection under management, and a lot of it is evolving by drift, and we're going to expect different genetic behavior there and levels of variation. We're working on this problem of lack of one-to-one orthology between model systems and crops between crops. And then this presence-absence variation and copy number variation is relevant to the whole field of quantitative genetics and population genetics, where we basically assumed everything is allele frequency changes over time, but actually the genetic basis of a lot of the traits we're talking about is not due to that. It's due to the number of copies of individual genes that we, again, lack some of the tools and theory to work with on. So thank you.
Okay, thanks. Now we have time for 1 or 2 questions.
So years ago, Scott Tingey did a thing where he said, B73 and Mo17 are as similar to each other as maize and rice. And there's been a lot of this, and you're throwing that out there. So I'm going to say, what proof do you have that those copy numbers are actually real and not just pseudogenizing and disappearing, and that a bunch of your synteny is, duplication, but who cares because the genes are dying?
Yeah, no, I didn't get into that on here, but I showed one example that the gene does something still, and many of them, 60% of them in any given inbred line of maize should be pseudogenes, is the average, but it's really difficult to distinguish pseudogenes from real genes, especially in maize and other crops where the most expressed parts of the genome are actually not genes, they're transposons. So we're, that's where this actually modeling pseudogeny, right, as a trait and treating it as a continuous variable is what we're trying to do. But because testing each one of these is not really feasible. You could study expression, you could study, you can do the sort of poly-A ribosomal enriched, but that's still, and you can study proteins, right? There's data on that. So all of that, from that, we know that many of these original whole genome duplications are retained and neo-functionalized. There's, data on that. But there's a huge area where we don't really know, and that was B73, right? And now we have, de novo assemblies and annotations of many inbred lines, and they will have a different set of genes which ones are actually pseudogenes and which ones are neofunctionalized. Yeah, I think it's going to be a computational approach and not an empirical approach.
Okay, we thank you once again for the nice presentation.